In this lesson, on our continuing tour through a cartoon cell, we're going to focus on the hereditary molecule for life on Earth, and that is DNA, with special attention to how DNA stores information. Now here was our cartoon cell. Here's DNA. In bacteria, it exists as a circular piece of DNA, circular molecule, sometimes called a circular chromosome. And we are already familiar with the basic structure of DNA being a double helix and how the molecule can be duplicated and can change when mutations occur. But we'll focus mostly on information in this lesson. In this scene down here, the cell is using DNA information to build a protein. So it's not surprising that to build a protein, you've got to have some instructions to know how to build it. Well, it's DNA that stores the instructions or the recipes to build proteins. The first step is that the DNA recipe will be copied into a molecule called messenger RNA. And then this pink structure here called a ribosome will read these instructions and build the appropriate protein. Now you can see up in our cartoon cell here there are many different proteins shown here. In fact, there are thousands of different kinds of proteins in typical bacteria. Here are just 4 of them over here. So we got the membrane transport protein, the channel protein, the protein that's going to break down glucose initially. We have the ATP production protein, and we have a molecular builder. Now you'll recall proteins are composed of amino acids, and there are 20 different amino acids. They differ by whatever is attached at this location to the carbon there. Proteins are composed of unique sequences of 20 different amino acids. So let's take a look at our 4 proteins over here on the right, and you'll notice here is the amino acid sequence for this protein, and here's the amino acid sequence for that protein. Notice they're different. In fact, all 4 proteins have a different sequence of amino acids. And that sequence is important because that determines how this chain will fold up, thus determining the 3-dimensional structure of the protein. And the structure of the protein determines the function of the protein. So these 4 proteins do different things because they have different shapes. They have different shapes because they had different amino acid sequences. Now every protein in the cell has its recipe stored at a unique location along the DNA molecule. So the recipe for this protein is stored in this section of DNA. The recipe for this protein is stored here, and this protein stored here, and this protein stored here. Now, how does DNA store information? So let's get to that. First of all, we've learned about the basic structure of DNA, and we know the rungs of the DNA ladder are composed of 2 bases, and there's a pattern to the pairing. An A chemical pairs with the T chemical, and the C chemical pairs with the G chemical. So we call this a double helix. It's a twisted ladder, and just 4 chemicals make up the rungs of the DNA ladder. Now we're going to untwist the DNA molecule, so we get a diagram like this. So we see the pairing here, and we've got the 2 strands of DNA just untwisted. Now we've learned about this stickiness that make the rungs of the ladder. The stickiness of the DNA bases is not how DNA stores information. The stickiness allows the DNA molecule to be duplicated when each strand separates, and then the spare bases can find their matching partners. So how then is DNA storing information? We need to turn our attention to the sequence of DNA bases along one strand of the DNA molecule. That's going to be a recipe. So, when scientists first started to understand the structure of DNA and the structure of proteins, they were led to this idea that since DNA and proteins are both chain molecules, then maybe the sequence of DNA bases specifies the sequence of amino acids in a protein. And in fact, this is the current understanding. It's a 3-letter code that the first 3 letters of this DNA sequence specifies the identity of the first amino acid in the protein. The next 3 letters specifies the identity of the next amino acid, and so on. So in a sense, what does DNA say? It says when the cell's ready to build a protein, it says start with this amino acid, then add that one, then add this one, then add this one, etc. So this stretch of DNA, the sequence of these DNA bases, is the instruction for how to link together the 20 different amino acids into some long chain that represents a protein. So a sequence of DNA bases along one strand codes for the sequence of amino acids in a particular protein. And we call this recipe for a protein a gene. So now we've got another vocab word. Each of the 4 proteins is composed of a sequence of amino acids, and that sequence of amino acids is uniquely specified by a sequence of DNA letters, and the sequence of DNA letters that codes for a protein is called a gene. Now, during the daily life of a cell, the DNA information is routinely accessed to build the proteins that the cell needs at that moment. So back to the scene here, here's the gene for this blue protein here. And the cell's going to make a temporary copy of that recipe in a molecule called messenger RNA. And then the ribosome is going to chug along 3 letters at a time. Each 3 letters of the messenger RNA specifies the identity of one of the amino acids, and then it'll build a chain of amino acids. It'll fold up, and then this protein will go off into the cell and do its job. Because DNA can be duplicated, the information to make proteins can be passed on to daughter cells when the cell reproduces. So here we have the parent cell, and because this DNA molecule can be duplicated, when the cell divides into 2 daughters, each daughter cell will get a full set of recipes to build all the proteins needed to keep the cell alive. Now, if the duplication of DNA is faithful and no mistakes, then the daughters are going to be identical to each other and to the parent. They should have similar traits. They should have the same traits. They will be clones. And this is what we mean by heredity, passing on of traits. So the reason why the offspring tend to look like the parents is because the parents are passing on the instructions that the parents were using to build the parent. And so the offspring are inheriting those instructions, so the offspring are going to look like the parents. Now, occasionally, there can be errors. So here we see the bacterial cell, and here's the circular chromosome, circular DNA molecule. When that DNA molecule gets duplicated, sometimes there can be errors that we call mutations. And so the daughter cells might actually be different from the parent and possibly different from each other. And these mutations then allow for the possibility of evolutionary change. So out of the 3 features of DNA, information duplication change, the fact that DNA can change its information content as mutations accumulate means that living things can change over time. And we're going to call that process evolution. Let's take a look at some bacteria in the process of dividing. Now, we're not going to be able to see all the biology going on inside the cell, but as you watch the video, you should imagine that the cell has already duplicated its DNA, and now we're going to see the bacteria cell split into 2 daughter cells. We'll call your attention to the one with the lightning bolt there. This cell has grown big, so it's sort of longer than the others. Right, like here's a small one, here's a larger one. So bacteria cells grow, and we've learned that they can't get too big or they face an energy shortage. This bacterium is in the process of dividing. So it has already duplicated its DNA, and now it's a matter of making sure each daughter cell has a copy of the DNA molecule and then dividing into 2 cells. Almost done, and there we go. 2 new individuals. Now, if there were no mutations in the duplication of the DNA, then these individuals would be clones, and they would have been just like the parent cell. If there are mutations, then these individuals can be different from the parent and different from each other.